Thank you for watching this edition of our video table of contents, or From the Editor's Desk. As usual, Volume 78, Number 2 for August 15th, 2015 has a lot to consume. This week we've published 2 reviews, 2 editorial focus pieces, 1 editorial, especially for young investigators, 17 original research papers, and 1 letter to the editor. I know you're anxious to take a peek inside, so let's begin. Our first review by Young et al. Discusses cardiomyoblast differentiation. Special emphasis is put on mechanoregulation of differentiation, parameters for evaluating the role of mechanical forces on 2D and 3D cell culture systems as well as in vivo systems are reviewed. In the second review by Fernandez et al., the effects of aerobic exercise training on microRNA expression and their role in cardiac remodeling is discussed. These authors provide an overview of studies from their laboratories and others showing the effects of exercise on the expression of pro- and anti-hypertrophic microRNA, as well as those related to vascular remodeling. You'll enjoy reading this informative review. Bender and colleagues assessed femoral pulse wave velocity in vivo and correlated it to isolated vessel function in a mouse model of Western diet, in other words, high-fat, high-sugar induced obesity and insulin resistance. They found increased TGF-beta content in Western diet-fed mice. Interestingly, coronary artery stiffness and structure was unchanged in Western diet-fed mice. These studies point out major regional differences in the development of arterial stiffness and dysfunction associated with a Western diet. Cordiero et al. Investigated the efficacy of BK calcium channel activation as a cardioprotective strategy during ischemia and reperfusion and the specificity of the BK channel activator Rotlin. Wild-type and BK channel knockout mice were subjected to an isolated heart model of ischemia and reperfusion. Adjunctive cellular experiments were also carried out. The results demonstrated that activation of BK calcium channels rescued ischemic damage associated with ischemia and reperfusion, likely by improved mitochondrial membrane potential and reduced ROS generation. Finally, Koijman and co-workers evaluated the effects of selective parasympathetic denervation of the spleen on inflammatory status and atherosclerotic lesion development in female ApoE3 Leiden mice, a model of atherosclerosis when fed high cholesterol. Selective denervation increased dendritic cells, B cells, and T cells in the spleen and increased gene expression of pro-inflammatory cytokines in liver and in leukocytes. However, this intervention did not affect the development of atherosclerotic lesions. I want to thank you again for watching this week. There really is so much more to digest in this issue, so please be sure to peruse the table of contents. Please write to me and provide me with your feedback, what you'd like to see in the journal. We have new and interesting content planned for the rest of the year. I hope to see you again in 2 weeks. Please send me your best work now.